So I'll start with some background around what NGF is and its role in terms of pain sensitization and peripheral nerve sensitization and signaling. Then we'll move into talking about NGF and inflammation. We obviously get a lot of questions around what NGF's role is in inflammation, so we'll speak to that. And then I'll move into some data that really does cover why we believe and why we can demonstrate that NGF does indeed have a role in various inflammatory pathways. And then I'll conclude with some upcoming studies or ongoing studies that we are working on in vivo, and then summarize the presentation. So next slide, please. So what is NGF? If you can move to the next one, thank you. So I'm sure everyone's familiar with nerve growth factor. NGF has been a target for pain research for decades, starting in human health and then shifting over to animal health. NGF binds to multiple receptors, but its primary target is the TrkA receptor. And that is defined as the high affinity receptor, and P75, which I won't really speak about today, is the other receptor, a low affinity binding receptor. Next slide, please. Once NGF binds to TrkA receptors, it forms a complex, a ligand receptor complex, which is then internalized into the dorsal root ganglion neuron, and it moves to the cell nucleus where it begins to work on transcriptional regulation. So this complex will alter the production of different proteins that are involved in pain signaling as well as inflammation through transcriptional regulation. As an example, some of these changes include upregulation of neuropeptides, which we know are involved in neurogenic inflammation. Two examples would be substance P and CGRP, which I'm sure many folks are familiar with. There are other mediators that it upregulates as well. The other thing that occurs is once these genes are transcriptionally regulated or upregulated, you get neuropeptide release back at the peripheral nerve ending and into the joint, which will then, of course, further increase inflammation as well as pain sensitization. Other transcriptional changes that can occur are the increase of neurotransmitters, of course, in terms of communication with the neurons, not only from the peripheral nervous system, but up into the central nervous system through the spinal cord and into the brain for perception. So shifting gears a bit around NGF's role in inflammation, if you can move to the next slide. So if we think the first set of slides were focused on the dorsal root ganglia and the neuron itself, but if we start to look at the knee joint and understand what is going on in terms of a very complex disease, of course, with OA, chronic OA, and if we take some very simple steps and look on the right-hand side of the schematic, it begins with some type of inciting event, and that can be a traumatic event, it could be a CCL rupture, something like that. What happens is you have damage to the cartilage and the synovial lining. That will begin a release of inflammatory mediators. Some of the common mediators you're well familiar with, like IL-1 or TNF alpha, those pro-inflammatory cytokines. The second step that occurs is once those inflammatory cytokines are released, you get a subsequent release of other mediators involved in inflammation, like prostaglandins and also nerve growth factor. From there, nerve growth factor, obviously, once it's elevated, will begin to bind to TrkA receptors, and it will start to drive those transcriptional changes in the DRG, as I mentioned earlier. Next slide, please. Once we have that upregulation and those transcriptional changes are underway, we get increased mediators like neuropeptides, as I mentioned, CGRP and substance P. That will lead to the neurogenic inflammation or inflaming of the nerves. Other things that occur in this complex disease are neuronal sprouting. As you can imagine, nerve growth factor is involved in generating new neurons or novel innervation into the joint, which will then, of course, drive more pain signaling and pain sensitization. Additionally, you'll have angiogenic factors that will be there to increase blood flow and increase support of those new nerves in the joint. And then most importantly, in terms of degradation, you get an increase in inflammatory mediators that are involved in cartilage destruction and synoviocyte destruction. Some players like matrix metalloproteinases will increase and begin to degrade the joint in terms of chondrocyte death and synoviocyte death. And of course, once that occurs, you have this cycle that continues because there are more inflammatory mediators released as chondrocyte death occurs. Next slide. So we have a pretty good understanding of what is taking place in terms of pain sensitization and pain signaling. This is a very complex schematic that's borrowed from Dr. Duncan LaSalle and his team. But you can see on the slide, there are a lot of mediators, including ion channels, neurotransmitters that are involved in neuronal communication, both peripherally and centrally, that occur. Next slide, please. What we really don't understand as well is the role of NGF in inflammation. And that's where I'm going to shift to speak and show some of the data that we continue to generate in our R&D labs here at Zoetis. So we're continuing to build an understanding and provide a focus on the role of NGF in various inflammatory pathways, including regulation of pro-inflammatory cytokines, upregulation of neurogenic inflammation mediators, and importantly, the increase of inflammatory degradation proteins that can be involved in chronic OA disease. Next slide. So to speak to this, to set this up a little bit, this is going to be all in vitro data that I'll present today. And we're using various cell systems to interrogate the role of NGF in inflammation. Very simply, 3 cell types that I've listed here as an example, and there are more, but just for the simplicity of the data that I'll present today, we use various neuronal cells. One is called a rat PC12 neuronal cell. And this is a cell line that's been involved in NGF research for decades. So in terms of understanding NGF signaling, it's a workhorse of NGF literature. We also use rat primary DRG neurons, and we stimulate those with canine or feline NGF because we get the same types of responses if we were to use rat NGF. It's a very homologous protein. And we also use joint cells, canine synovial-derived cells, to look at what is going on in terms of NGF regulation of those types of cells in the joint. What we're looking at least initially, is to understand transcriptomic changes as well as proteomic changes. Simply put, what genes are upregulated or downregulated upon NGF stimulation, and therefore what proteins are upregulated or downregulated once NGF stimulates these cells. And importantly, we're also looking at what is the response when we treat these cells with NGF plus our monoclonal antibodies like Librela and Solensia. So in a simple system, you can see that, thanks Stephanie, you can see at the bottom, we'll have an untreated cell type, which gives us our baseline. NGF will show the changes that we're looking for, and then we're looking for that blocking or inhibiting of those changes. Next slide. So anyone that's worked on transcriptomics or proteomics knows you get a lot of data out of these types of studies. Really the first step is to use a lot of different software tools to cluster the genes that are changing in these various experimental conditions. And what you can see in the blue box is a list of genes based on functional families. And I've highlighted within that blue box some red boxes that highlight why that NGF is increasing protein families or gene families that are involved in inflammation, like lymphocyte activation, cytokine production, as well as positive regulation of cytokine production. And to give you some context of what all the circles mean, essentially, the circles, the larger the circle, the more genes are changing within that gene cluster or that functional family. The colors represent whether it's an upregulation or downregulation. So red means upregulating, and blue means downregulating. Obviously, you can have some genes within these functional families that can go either way, and so that tends to show up as purplish, a mixture of those colors. Now, to dig in a little further, if you build the slide for me, Stephanie, and focus in on, if we dig into one of those families and look at the proteins or the genes in that family. One example here is an IL-4 receptor gene expression change. Essentially, what we're showing is that when we apply our monoclonal antibodies, 2 different ones at the bottom, ZTS768 and Solensia, it's NVO2, which is just our internal nomenclature, we get an increase in IL-4 gene expression. That's shown in the blue and the red bars. The difference really is that the red bars are 22 hours post-treatment and the blue are 7 hours post-treatment. So it's just a difference in timing. We obviously get more gene changes over time. And if you build one more, the importance of this is that IL-4 is a well-known anti-inflammatory gene or a protein, and it has been shown to have chondroprotective effects. So at least in this instance, it's one way for us to start focusing in on genes that we think have an important role in either anti-inflammatory or pro-inflammatory effects. And in this case, our mAbs increase an anti-inflammatory protein in this cell system. Next slide, please. To take it one step further and start to dig into more inflammatory mediators, I've bucketed these by what I consider 3 different domains of inflammation. So on the left-hand side, we have pro-inflammatory mediators. In the middle, neurogenic inflammation. And on the right-hand side, inflammatory degradation. So starting on the left-hand side, what we're doing is stimulating, again, these cells with NGF and looking at the gene changes as well as protein changes downstream. And we can see that we can show NGF upregulates phospholipase A2, which, as folks may remember, is an arachidonic acid synthesis, a precursor to prostaglandin production. It also upregulates COX-1 enzymes, which again is important in synthesis of inflammatory prostaglandins. And then something a little bit different outside of the NSAID or the prostaglandin pathways, we see an upregulation of IL-17 receptor, which is a pro-inflammatory cytokine that has been linked to arthritis in the literature. Shifting to the middle, I mentioned earlier that we know that NGF upregulates neuropeptides, and we've demonstrated that again with CGRP upregulation, as well as tachykinin 4, which is a cousin, if you will, of substance P. On the right-hand side, and this is where it's really important in terms of joint degradation, NGF is upregulating matrix metalloproteinases, in this example, MMP10 and 13, which we know are important in breakdown of the extracellular matrix and chondrocyte destruction in the joint, as well as other families that are related, like ADAMTS. And so the important aspect is not only that NGF is upregulating these genes in this cell system, it's that when we apply our mAbs to this experiment, it inhibits all of these NGF-mediated changes. Now, shifting a little bit to focus on one particular class of proteins, the matrix metalloproteinases, or MMPs for short. On the left-hand side, you can see a graph where we are measuring gene fold changes, and you can see that NGF in these cells upregulate, and that's in the orange, by almost 8-fold, or a little over 8-fold, gene expression change when you apply NGF. On the right-hand side of the graph, it's a little hard to see, but you can see that Librela and Silencia applied to the system inhibit those changes as well. So again, NGF upregulates MMPs, and our mAbs can block those changes. On the right-hand side in the table, it's just a lot of other MMPs. There's a lot of subtypes of this protein family. And you can see if you look at the column with NGF, we're increasing most of these anywhere from 2 to 8-fold in terms of gene expression, which is a very large change in gene expression. And the right side of the columns are Librela and Silencia treatment, which show that we are inhibiting NGF-mediated changes in MMPs. Now, the important aspect of this is that we submitted this data to the US FDA, or CVM, the regulatory agency that approved Librela, and they put this type of language that is shown below onto the label. In other words, they use these data to confirm that, yes, NGF does have a role in upregulating these inflammatory mediators, as is written down below, and that our mAbs decrease those changes, and that's actually on the labels of the product. Next slide. So shifting a little bit to more functional data. So I've been talking about gene changes and protein changes, but what's happening in terms of function of these neurons? We can measure electrical activity of these neurons, not quite through electrophysiology. We have a separate system called the Axon Maestro, which does measure stimulation of the neurons. And we can show that when you plate DRGs from rats and look at what is taking place electrically. What's the communication of these neurons, so to speak? What is occurring when we add NGF, and then what is happening when we inhibit with the mAbs? The orange bar shows that NGF is going to dramatically increase the signaling and electrical activity of these neurons as we measure, they're called spontaneous spike recordings. And then you can inhibit those changes with Silencia and Librela. So now we're showing the function of the neurons are excited by NGF and inhibited, which makes sense because, of course, when they're excited, you're getting more pain signaling, more peripheral sensitization, and the mAbs are dampening that down. Now, on the right-hand side, what we've done is we've looked at the function in these DRGs and then taken RNA from the same experiments to understand what is happening at the gene transcription level outside of the function that we measured. And you can see, again, some familiar proteins or genes in this case, CGRP and substance P. We get an upregulation of both of those, as we've shown before, but in this different prep, this DRG prep. And we're also showing that you get an increase in ion channels, which of course is important in terms of communication of the neurons and signaling. In terms of depolarizations or increases in signaling of these neurons, we get, as an example, an increase of over threefold of TRPV1, which is the capsaicin channel, and then NaV 1.7, which is a sodium channel that's involved in pain and has been in the literature for decades as well. So it all makes sense that we're getting this increase in the transcription of these ion channels, which is translating to an increase in function of the neurons themselves. And the most important part on the right-hand side is that when we add the MABs, Silencio or Librela, we decrease those changes both in terms of transcription, but also in terms of function of the neurons. So we are quieting the neurons, so to speak, that we believe is what's leading to the pain decrease or analgesia. So it's important to note that even though you see gene changes, that doesn't always equate to protein changes. So it's good to confirm that you are also seeing protein changes in your experimental systems. So here we're looking at DRG neurons, and we're looking at a variety of different types of proteins. We're looking at other neurotrophins like brain-derived neurotrophic factor, BDNF, which we know is important in pain signaling. We're looking at CGRP again, involved in neurogenic inflammation. And we're looking at neurotransmitters like glutamate, which we know is very important in terms of translating that signal from the peripheral nervous system into the central nervous system and the perception in the brain. And also VEGF. I mentioned earlier angiogenesis being an important part of disease, and we're showing that VEGF is also increased by NGF. All of that is shown on the right-hand side in the graph, where you have untreated in the first set of bars, and you can see the colorations. Gray is BDNF, red is CGRP, blue is glutamate, and green is VEGF. When you add NGF, which is the middle part of the graph, you can see all of those bars increase. That means we are getting increase in protein and all those different types of proteins that are key pain, neurogenic inflammation, and angiogenesis proteins. And then again, it's very important that we're showing with Librela. When you add Librela application to this experimental design, we bring that back down to baseline. We've inhibited those NGF-mediated changes in these key proteins that are involved in pain and inflammation. Next slide. So shifting one more time away from DRGs and neuronal cells and now looking at joint cells. So these are synoviocytes that are canine derived. And what we're looking at in our next set of experiments is to understand what is happening in terms of the inflammatory mediators as well as NGF in those synoviocytes. On the left-hand side, the graph is showing that IL-1 beta induces gene expression changes in these canine synoviocytes. And one of the key gene changes that you can see if you look at the orange bar is NGF. So we're demonstrating that we have a system that mimics what we believe is occurring in the joint itself. That is, a pro-inflammatory cytokine is released like IL-1 beta, and it begins to upregulate NGF, as well as a bunch of other mediators like PLA2 and MMPs, as well as TRPV1 in the same system. The right-hand side of the table just shows the numbers that are associated with the graph. And again, you can see the increase in gene fold changes across those different types of genes that are involved in OA disease. What we're now going to do is understand whether the MABs can blunt these effects because we're not acting on IL-1 beta, we're acting on binding to NGF. So can we reduce some of these gene changes by applying our MABs? And that's some data that we're still working on in our labs as we speak. Next slide. The last data slide that I'll show is really more of a representation of what we're working on in vivo. So all of the data that I've shown today is in vitro data, and that's very important because it gives us an understanding of what genes and proteins to look for in terms of what NGF can do in terms of gene regulation or protein regulation or even function of the neurons. But now we're working to translate that to rodent models and canine models. As an example, rat MIA, which is a very well understood and documented OA disease model in rodents. Essentially, it's a intra-articular injection of monoiodoacetate, which will destroy the knee joint, and that's pictured below on the right-hand side. You can see that over time, if you look at MIA at 11 days or 28 days in these IHC pictures, that you get a decrease. You can see in the middle top blue, we have some red arrows. Hopefully that's coming through. That's basically depicting that the cartilage and the synovial lining are thinning over time because of that MIA destroying of the chondrocytes. And one of the studies that we have underway, both in rodents and also canine, is to understand when we have that type of joint destruction, if we apply the MABs early on in that process, are we able to slow that disease progression, at least in laboratory models? And we hope to have that data within the first half of this year to really be able to start understanding the translation of what we see in vitro to in vivo. Next slide. So in conclusion, hopefully you'll have seen that we are really focused on building an understanding of the role of NGF in various inflammatory pathways, whether that be pro-inflammatory cytokine regulation, neurogenic inflammation, or inflammatory degradation. Currently, our in vitro data is very conclusive in that clearly demonstrates that NGF upregulates a wide variety of inflammatory mediators in both rat neuronal cells and dorsal root ganglion neurons, which are important in pain signaling. And then our MABs, both Librela and Silencia, or Benvetmab or Frunevetmab, inhibit these changes, these NGF-mediated changes. Next slide. I won't rattle off all the genes, but just so this is moving a little bit fast, so you can see again what we saw and what I showed earlier in terms of pro-inflammatory cytokines. You see some of those common players that are involved in prostaglandin production. Those are upregulated by NGF in our experimental systems and inhibited by our MABs. And then, of course, the neurogenic inflammation peptides, neuropeptides that I've shown several times. And I think most importantly, the inflammatory degradation proteins, the genes that are involved in upregulating those proteins are increased by NGF, and we can block those with the MABs. Clearly, Librela and Silencia can inhibit very robustly these NGF-mediated changes in these pathways, and will continue to drive experimental designs that help build that translation between in vitro to in vivo to really show what the potential is for these MABs to hopefully slow down the progression of a very complex disease. And hopefully the next time we do this, we'll have that type of data to show the in vivo studies. Thank you.